Liver. This organ assures that a constant supply of fuel is available to tissues, is central to the maintenance of metabolic homeostasis, stores glucose to glycogen, to Fa to triglycerides, stores cholesterol, lipoproteins, albumin, bile, drug detox, excretion. Insulin. Polypeptide containing a total of 51 amino acids, stimulus blood glucose produced b cells of the pancreas. Functions: 1. Glucose uptake into muscle, adipose tissue, 2. Energy storage. Decrease in blood glucose. Release of glucagon from cells of pancreas. Release of epinephrine from adrenal gland. Epinephrine stimulates release of glucagon from cells. Increase in blood glucose. Secretion of insulin from B cells of pancreas. Crosstalk. Insulin inhibits secretion of glucagon from cells. Insulin inhibits glucagon gene transcription. Glucagon. Polypeptide containing 29 amino acids, stimulus produced, cells of the pancreas. Functions: gluconeogenesis, release of FAs from adipose, and FA oxidation. Glucose balance is especially important for brain fuel. Crosstalk. This interaction between hormones involves blood glucose releases insulin, which inhibits glucagon secretion from alpha cells, and glucagon gene TXN, or blood glucose releases EPI, from adrenal gland. This can stimulate release of glucagon. Adenylyl cyclase. This enzyme is triggered by glucagon. It synthesizes CAMP. Specificity depends on tissue localization of glucagon receptors. 1. Glucagon receptors are present in liver and adipose tissue. 2. Muscle lacks glucagon receptors. CAMP. This second messenger is synthesized by adenylyl cyclase, and it then activates pKa. pKa. This enzyme is dependent upon CAMP. Once activated by CAMP, it will activate PDE through phosphorylation. This enzyme is primarily responsible for mediating phosphorylation in the glycogen response. PDE. This enzyme degrades the phosphodiester bond in CAMP and cGMP, thus, they regulate the localization, duration, and amplitude of CAMP and cGMP signaling. Consequently, they are important regulators of signal transduction mediated by these second messenger molecules. A kinase anchoring proteins. These proteins help localize pKa. They bind to the pKa regulatory subunits, catalytic regulatory and also bind, anchor to a specific cell structure, they do not affect pKa activity. Gs. This protein is activated by glucagon or epinephrine. It then activates adenylyl cyclase, which then synthesizes CAMP. CAMP then activates pKa, which phosphorylates proteins of the serine, Threonine. AKT. This kinase is primarily responsible for mediating phosphorylation in the insulin response. Upstream signal from glucagon or epinephrine. This action will regulate CAMP levels by activating ACT, which converts ATP CAMP. pKa negative feedback. This action will regulate CAMP levels by phosphorylating, activating PDEs, which degrades CAMP. Insulin-activated kinase inhibition. This action will regulate CAMP levels by activating PPP, which phosphorylates, activates PDE. PPP. This is a family of enzymes with conserved catalytic subunit and variable regulatory subunit. The enzyme is activated by phosphorylation of the regulatory subunit, the liver phosphoprotein. Phosphatase acts on proteins that are phosphorylated on serine or threonine, turns CAMP signal. Insulin-activated kinases. These enzymes activate PPPs, which can then remove Ps from proteins. One example is GSK3, which phosphorylates PP1. These enzymes also activate PDEs, which degrades CAMP. In sum, these enzymes oppose the actions of glucagon or epinephrine. PP1 inhibitor. This molecule is important for when you want pKa to work. You want to prevent PPP from doing its thing. This molecule is stimulated by CAMP, pKa-dependent phosphorylation. The activated molecule inhibits PPP. Consequently, the pKa and other proteins stay phosphorylated and stay active. Keeps CAMP signal on. What are types of hormones? Endocrine hormones synthesized by endocrine tissue and carried in the blood to another tissue, where it acts as a messenger to regulate the function of the target tissue or organ. Paracrine hormones are released into the EC space and diffused to neighboring target cells. What are examples of endocrine hormones? Insulin and glucagon. These have a range of to m. What are examples of paracrine hormones? Eicosanoids, nitric oxide. These are short-range. What are autocrine hormones? do not require external stimuli, and they act on the same cell through the receptor. Neuronal signaling. Neurotransmitters are released that act on nearby cells. Endocrine signaling. Endocrine glands release hormones that act on cells far away. Stimulation of glycogen breakdown, stimulation of growth and differentiations, regulation of water retention by kidneys. Five types of direct events following hormone binding. 1. Second messenger is released inside the cell. 2. A receptor TYR kinase is activated. 3. A hormone gatadiene channel is opened or closed. 4. An adhesion receptor send info to cytoskeleton. 5. A steroid bound to receptor protein in nucleus alters gene expression. Endocrine targets. Direct target equals anterior and posterior pituitary. Anterior produces tropins and posterior produces oxytocin and vasopressin. Secondary targets equals adrenal cortex, thyroid, ovaries, testes. Tertiary A equals tissues, organs. What hormones does the hypothalamus produce? Oxytocin, vasopressin. What type of organ is the anterior pituitary? It is an endocrine organ that received releasing factors from hypothalamus via blood vessels. What causes the release of anterior pituitary hormones? Releasing factors. What causes the release of the posterior pituitary hormones? Neuronal signals. What is cortisol derived from? Cholesterol. Where is cortisol produced? Adrenal gland. What is stimulated by? Corticotropin hormone from the anterior pituitary gland. What are the targets for cortisol? Muscles, liver, and adipose. What does cortisol do? Restores blood glucose, and restoring glycogen stores. Metabolic role is to counterbalance insulin. What type of hormone is insulin? Peptide hormone. What type of hormone is epinephrine? An amine hormone. How does epinephrine increase energy? It activates adenyl cyclase that then produces CAMP, then CAMP activates CAMP-dependent protein kinase, which activates glycogen phosphorylase B kinase. This then activates glycogen phosphorylase B. That breaks down glycogen to glucose 1-phosphate for energy. What type of hormones pass through plasma membrane? Nonpolar are like steroids, retinoid, vitamin D, and thyroid hormones. These bind to receptor in nucleus, where it binds to DNA and acts as a transcription factor. What type of hormones work faster? Peptide and amine hormones work faster than steroid and thyroid hormones. Insulin. Peptide hormone made in pancreas by beta cells. In muscle it facilitates glucose uptake. In liver, it promotes glycogen synthesis. In adipocytes it promotes glycerol synthesis and inhibits breakdown of fats. How do sulfonylurea drugs work? By stimulating insulin secretion, it prolongs the depolarization, causing the exocytosis of insulin. Glyburide and glipizide equals ATP-gated K-channel blockers. What causes insulin to be released? Increasing blood glucose levels. What is the C-peptide of insulin? It is a part that is cleaved off from proinsulin by proteases to make the insulin active. The protease activity is regulated by circuit 2. How does glucagon raise the blood glucose? In the liver it activates glycogen phosphorylase and inactivates glycogen synthase. It reduces F2,6-BP, so it inhibits glycolysis and stimulates gluconeogenesis. It inhibits pyruvate kinase, leading to low ACOA, which means fat synthesis being blocked. Accumulation of phosphoenolpyruvate favors gluconeogenesis, stimulates PEPCK for gluconeogenesis. How are lipid droplets exposed to lipase? By phosphorylation of paralipine. What happens when you are fasting or have uncontrolled diabetes? It will lead to lack of oxaloacetate that prevents the entry of ACOA into the CAC, so it starts to accumulate. The accumulation of ACOA leads to ketone synthesis, leading to the transport of them to the brain to be used for energy. This leads to ketosis. What is considered high fasting blood glucose? 126 or higher. What is considered a low fasting blood glucose? Below 50 in men or 40. Types of diabetes. Type 1, insulin deficiency, due to destruction of beta cells. Type 2, insulin resistance, due to defective insulin receptors. Treatments for diabetes. Type 1, administer insulin when glucose is rising. Type 2, weight loss and increased physical activity. What does high blood glucose increase the risk for? Cardiovascular disease, renal failure, damage to small blood vessels and nerves. Treatment for type 2 diabetes. TZDs-act on PPAR-gamma. It stimulates expression of genes, potentiating the action of insulin in the liver, muscles, adipose tissue. It increases glucose uptake and decreases glucose synthesis in liver. Biguanides act on AMPK, increases glucose uptake by muscles and decreases glucose Production in liver. GLP-1 modulators acts on glucagon-like peptide, and it enhances insulin secretion by pancreas. Leptin in control of body mass. It tells the brain that no more food is needed. It also stimulates production of anorexic peptide hormones. It also regulates the sympathetic nervous system, increasing blood pressure, heart rate, and thermogenesis.